Hi, good evening, everybody. My name is Danielle McDonald, and I'm a professor of marine biology and ecology here at the Rosenstiel School. I'm also the scientific director of the National Aplysia Resource. It's my pleasure to welcome everyone on campus and virtually to the second lecture of the 30th anniversary season of Sea Secrets. Pretty amazing, right? Our public lecture series was started by former Professor Robert Ginsburg and has been expanded over the years to include Rosenstiel scientists and other leading experts in marine, atmospheric, and earth science. At this time, we'd like to take a moment to thank our Sea Secrets sponsors. So, presenting sponsor Norwegian Cruise Line Holdings Limited and the Shepherd Broad Foundation, William J. Galway III, Cheryl Gold, KB Life Enhancement Forum, Key Biscayne Community Foundation, Joan McCon Family Foundation, Nicole and Myron Wang, and Southern Glazer Wine and Spirits. Before we start today, and before I introduce our awesome speaker for this evening, I would like to talk to you a little bit about a new center we have here at the Rosenstiel School, the Glassell Family Center for Marine Biomedicine. I'm currently the director of this center, and I'm just going to take a few minutes to talk about this exciting expansion of the Rosenstiel School's mission. So you may be wondering, marine biomedicine, what is that, and why would a marine school be doing anything associated with biomedicine? Well, in fact, the Rosenstiel School has been in the business of human health for a long time. So Rosenstiel atmospheric scientists and chemists are experts in measuring the changing environment, and these pose risks to human health. So things like increasing temperatures or the idea of urban heat islands, things like algal blooms, atmospheric pollution, water pollution. All these things pose risks to human health. Rosenstiel social scientists are also experts in measuring health risk. So they're looking at things like the impacts of climate or the impacts of socioeconomic status on human health risk. For example, how climate or socioeconomic status can increase your risk to cancer. Rosenstiel marine biologists use marine organisms as models to look at the impacts of environmental change, like heat or pollution, and also to model human diseases. So up here we have the bicolored damselfish, for example, right here has a natural cancer. So we can use that animal to study certain cancers in humans. The sea anemone larva can regenerate. We can use that as a model to study controlled cell growth and compare it to what happens in cancer, which is uncontrolled cell growth. Rosenstiel marine biologists also grow heart and brain healthy food. So our scientists are looking at how to effectively and efficiently grow fish and how to make them even more healthy for us. So UM is in the unique position to have a medical school and a marine school in really close proximities. But even though we're only 7 miles apart, sometimes the distance is even greater, and feels greater, and that's because, the medical scientists at the Miller School and the marine scientists, atmospheric and earth scientists at the Rosenstiel School, have their own projects to be worrying about. So part of my role and part of the role of this new Glassell Family Center for Marine Biomedicine is to help connect the Miller School and the Rosenstiel School, improve collaboration, And that will lead to innovation and discoveries in human health science. The goal of the Glassell Center is also to bring in other schools here at the University of Miami. For example, engineering, as well as the School of Nursing and Health Studies. These two schools and colleges also have, do a lot of human health research. So through this interconnection and increased collaboration, we will be able to innovate and work to advance discoveries in treating the world's leading diseases such as heart disease, cancer, stroke, and health factors such as aging, pollution, socioeconomic disparities, and climate change. So, one thing that we don't do here at Rosenstiel and would love to be doing more of is looking at marine natural products. And that is the topic of our speaker tonight, Dr. Esther Guzman. So it is my pleasure to introduce Dr. Guzman. Dr. Guzman received her Bachelor of Science in Molecular Biology and Biotechnology from Salem Teikyo University and her Ph.D. in Biomedical Sciences with emphasis in immunology from the University of Texas MD Anderson Cancer Center in Houston. She leads the Cancer Cell Biology program at Harbor Branch Oceanographic Institute at Florida Atlantic University. For the last 2 decades, she has been focused on finding marine natural products that have the potential to be novel therapeutics against the over 100 diseases grouped under the term cancer. These include using marine natural products to prevent cancer formation, decrease inflammation, eliminate existing tumors, reactivate the immune system to fight cancers, stop tumors from spreading to other organs, or use as tools to further our understanding of cancer. Esther, welcome to C-Secrets.
Thank you. Thank you so much for that kind introduction and for the invitation to be here. I always like to start with this slide because everybody has the question, why explore the oceans for medicines? And basically, marine natural products or secondary metabolites are compounds that get made by marine organisms that are not essential for their survival, but it gives them an advantage by having them. And so when you see a beautiful reef like this, you can see how many organisms are living side by side and sometimes on top of each other. And these organisms cannot move. So in order to defend themselves, they must make natural products to kind of fight off the invaders. But the other super cool thing about this is natural products are inherently bioactive. They were made by nature for a reason. Now, I don't understand what the reason is that a sponge needs a compound that serves human cancer, but they obviously have activity. And so if you see these statistics, over 62% of small molecules approved currently as medicines can be traced back to natural products, and over 48% of cancer medicines can be traced back to natural products. And this is in the majority from terrestrial natural products. So the oceans that cover 70% of the Earth must have amazing natural products that are waiting for us to discover them and put them to good use. And of course, we're in Florida, which has the second longest coastline, and so we have those amazing resources right here in our backyard. And drugs from the sea are slowly becoming a reality. So I put structures in case there are chemists, because they always go and complain that the mean biologist doesn't include the structures. So here you have them. And this is cytarabine, which has been approved since 1969 to treat leukemia, and it comes from a Caribbean sponge. Halaben, or eribulin mesylate, is the synthetic analog of the natural compound helicondrin B that comes from Helicondra orchidae, another marine sponge, and has been approved since 2010 to treat metastatic breast cancer. Jondalis, or actinicidin 743, is the semi-synthetic version of a natural product that comes from a tunicate and has been approved since 2015 to treat soft tissue sarcoma and ovarian cancer. And there's a new type of drug that is all the rage right now in cancer treatment. And these are what we call antibody-drug conjugates. And they combine a payload, which is usually a natural product that kills cancer cells, with a linker to an antibody. And that antibody will direct it to the site of the cancer. So the antibody will get it there, the linker will break, and then the payload will kill the cancer cells. And so this specific linker with this specific payload is called commercially or clinically as vedotin. And it uses monomethyl auristatin E, or MMAE, which is the synthetic version of a natural compound that is made by a cyanobacteria, and it kills cancer cells. And this one has a funny story because it was first isolated from a mollusk, and then they figure out that it's a cyanobacteria that makes it. But combinations of this linker and payload with different antibodies have been approved since 2019 to treat lymphomas, to treat metastatic urothelial cancers, and since 2021 to treat metastatic cervical cancer. So you're going to be hearing a lot more about this because it is one of the hottest topics for cancer treatment right now. But of course, it's not everything as beautiful as we have. So you heard that I've been working on this for like 20 years, and you're like, so where are your drugs? Well, it takes a long time for a compound to make it to a clinic. So this is the story of actinium 743. This is a beautiful tourniquet, and it grows like right in Fort Pierce, right where we work. But in 1969, an extract was found to have anti-tumor activity at the National Cancer Institute. It took 21 years for somebody to figure out the structure of this compound. It happened at the same time by Reinhardt et al. At the University of Illinois, Urbana-Champaign, and by our very own Dr. Amy Wright at Harbor Branch Oceanographic Institute. And she just got a big award for that structure. And that is a complex structure. And the reason that is important is if you want to make the compound in the lab, you need to know the structure. But even knowing the structure, it took PharmaMar another 10 years to report a commercial synthesis pathway. And that just means a cost-effective way of making it in the lab. And so 10 years for that, another 7 years for the European agencies to approve it for advanced soft tissue sarcoma, and another 8 years for the FDA to approve it in the US. So it's a long road. We're hoping that it is not as long as this, but this is the reality of what happened with this particular compound. And so I'm part of the Marine Biomedical and Biotechnology Research Program at Harbor Branch Oceanographic Institute. And one of the things that has characterized this program since its inception in 1984 is the multidisciplinary approach. So we have natural products chemistry, collections, microbiology, and what unites us all is the desire to discover marine natural products that are useful to treat dreaded diseases. So we find antibiotics, we find anti-cancers, and we collaborate with other groups to do more work. My group is obviously the cancer cell biology, so everything's going to be skewed to that, but I just wanted to introduce you that there's other stuff happening there. And so what is cancer? Cancer is a collective name for over 100 diseases, so there will never be one single cure for cancer. And it's always hard to say it, but that's the reality of it. What they have in common is that you have cells in part of the body that are growing uncontrollably or failing to die, and they have damaged DNA. And how that damaged DNA happened could be from inheritance. You got those genes, you were exposed to chemicals, viral infections, smoking, excessive exposure. But one thing to remember is having a mutation doesn't mean that you will have the cancer. So we have what we call the two-hit theory, in which more than one event must happen before cancer develops. And the good news is our fight against cancer has been successful. Between 1991 to 2021, there's been a reduction of 33% in cancer deaths. And this is due to reducing the amount of smoking that happened and advances in early detection and treatment. And these advances in early detection and treatment happen because people invested in cancer research. The more we understand a disease, the better we can fight it. Unfortunately, the fight is not over yet. So cancer remains the second leading cause of death in the United States and in Florida. We expect over 2 million new cases of cancer in the US this year, with 600,000 deaths. And in Florida, it will be about 172,000 new cases, and about 49,000 deaths. And part of the problem is we're living longer, which is a good thing to have, but because we live longer and the risk for cancer increases as we get older, then we're seeing more cases than we used to see in previous decades. So it's a trade-off. Of the deaths that are going to occur, 21% will be due to lung cancer, 8% to colon cancer, 8% to pancreatic cancer, and about 7% to breast cancers. So I'm very lucky that I work at Harbor Branch Oceanographic Institute, and for many years we had our own ship and submersible that allowed us to collect marine invertebrates. So I got to see the bottom of the ocean with my own eyes. It was pretty amazing. Now we use remotely operated vehicles to do the same thing, and we collect marine invertebrates, and then we extract with solvent, and I'll tell you what that is in a second. And we do fractionation or separation of the compounds, so you can assume that every little peak that you see is a compound. And those get separated into mixtures that have 3 or 4 compounds to create what we call our peak library. And of course, we isolate pure compounds. And so we have those 2 libraries that are sent to my group for screening for activity. And if we have an activity, then we go into how does it work, or mechanism of action studies. And so how do we make an extract? If you are like me, you depend on this every day to get you going. So anytime that you're making coffee or make tea, if you are a tea drinker, you're making an extract. You're basically grinding your coffee beans or taking your tea leaves and mixing it with hot water, and that extracts the chemicals. And so when you get this fantastic drink, what you're getting is caffeine and about 1,000 other chemicals that give it its flavor and its smell. And so, yeah, you can tell that I drink a lot of coffee, right? Our chemists follow a similar approach. So they grind our little organism, whether it's a sea fan or a sponge. They use alcohol instead of hot water, and chances are you don't want to drink this, so I don't recommend them. But you can see the extracts here, and unfortunately, those that look super beautiful, they're never active. So why do we use the peak library to find compounds? As I said, those are mixtures of 3 or 4 compounds. And the reason we use that approach is that this is a Discodermia dissoluta sponge. And it contains compounds that are very, very cytotoxic at very, very small quantities. If we use a crude extract, that's all we would see, those very cytotoxic compounds, and we would miss everything else that is within that sponge. Because we use the peak library, though, our chemists were able to follow the activity. So you can see the green activity, and that led to this compound. And this compound inhibits IL-8. So why does that matter? Well, IL-8 is a soluble factor that is produced by cells, and it helps to create new blood vessels. So you don't want cancers to have that, and by inhibiting that ability, you're actually fighting cancer even though you're not killing the cancer itself. Another way that we do this is this, following the yellow activity, we found a compound that has CCL2 inhibition. That's another soluble factor, is what we call a chemokine, because it attracts cells to a site where it's being released. And so CCL2 attracts cells of the immune system to pancreatic cancer and other cancers, and makes them release stem cell factors and factors that help the tumor grow. So by preventing those cells from getting that, we take away one of the initiating events on that particular cancer. And so you can fight cancer, not necessarily killing the cancer cell. So one of the things that we use in my lab to study this is what we call cancer cell lines. So if a patient goes to surgery to have a tumor excised, this tumor is dissociated or separated into what we call single cell suspension, and scientists try to grow it in the lab. Now, this is not a trivial process. It takes a long time. Not every cell will take. But if they take, they're placed in repositories such as the American Type Culture Collection, where scientists like me can buy those cell lines. And the advantage of this is that those cells have all the mutations that led to the tumor being formed, all the soluble factors that the tumor had, and all the characteristics of the cancer. So I basically get to test in humans without harming anybody. And so it's a beautiful way of doing science. I'm going to introduce you to a new technique. It's called high-content imaging. We use this a lot in my lab, and so at some point I'm going to show you results that had to do this. And this is kind of like the general approach that we follow in my lab. And high-content imaging is a specialized microscope that takes pictures insanely fast, but it also can quantitate the effects of a particular fluorescent tag. And so it allows us to not only my naked eye saying, I think that's reduced, but actually have numbers and saying this effect happened. So this is my general approach. We plate cells and allow them to adhere overnight. We replace with media containing our marine compounds or controls. We let them sit for about 24 hours, sometimes it's more. Then we fix them. And fixing means adding a chemical that is basically, if you guys play freeze when you were a kid, freeze. That's what we're doing to the cell, We're making it stop exactly at the point that whatever our compound did to them and capturing that so that then we can use fluorescent tags and see what's happening to the cell and seeing what effect our compound had. And so we get things that look like this. So these are cancer cells. Our target is in red. You can see that when we treat with a natural product, you see a lot less red. And so that's kind of some of the ways that we're looking at data. The other important thing about high-content imaging is that it allows us to test a lot more things a lot faster. So in a test tube, you can do one test, and that's it. We used to use 96 well plates, and so you can think of every little circle as a test tube. So now we have 96 test tubes, and we can test 48 samples in duplicate. But the high-content imager can use 384 well plates. So now every little square is a test tube, and now I can test about 192 samples in duplicate at once. And so for me, if I wanted to screen about 1,000 samples with a 96 well plate, it used to take me about 6 months. If I want to do it with the high content, I can test 3,000 samples in about 4 months. And so the throughput is just so much higher and allows me to get more results faster. I'm going to introduce you to a couple of concepts that are important in cancer biology that I know I'm going to mention at some point. And so the first of it is apoptosis. And apoptosis is a Greek word that stands for programmed cell death. And so it is a very normal process by which cells that have finished their function, that have been damaged in any way, that are not necessarily, that we don't need anymore, are eliminated. And one easy way to think about this is when you have an infection and somebody takes blood, and they tell you have elevated white lymphocytes. That's how we know you have an infection. And they know you're true with the infection when they test you again, and those levels are down. And so what happens is when the immune system is fighting, you get a lot of the white lymphocytes going. When they finish their function, they die through this process because they're no longer needed and they can go away. And so apoptosis is a very important system in the body. And unfortunately, many cancer cells are resistant to apoptosis. So they get the signal that they should be dying, but they don't die. And now I'm going to introduce another system, another concept, which is signal transduction. And signal transduction is what happens inside your cell. And the easiest definition is the way that you transfer one signal or stimuli into another. But the best way to think about this, I was trying to explain this to a chemist, and they came up with this symbol, and I liked it, so I'm going to use it. So if you order a pizza, the stimuli, it's going to get translated into the delivery guy getting into his car, and if they follow all the traffic signals, you get a yummy pizza. Great outcome, this is great. However, if the delivery guy gets into his car and encounters one light that is not working, So you're going to get into an accident and no pizza. So same outcome. So one easy way to think about this is normal cell, all signals work, we have pizza, great. Cancer, something is not working with that signal, and so you get a crash, you get something bad, no yummy pizza. And so what we do in my group is we take marine natural products, we put them against human cancer cells, and we try to see different ways that they can cure cancer or treat cancer. We start from killing cancer cells. Of course, we don't just simply want to kill cancer cells. We want to kill cancer cells without killing normal cells, because anytime that you see anybody going through chemotherapy, if they're losing their hair or they're throwing up, it's because normal cells are being killing, sometimes at very high rates. And so we're looking for those that have that selectivity. They kill many cancer cells, thousands of cancer cells for each normal cell that they kill to eliminate those side effects. We're looking for things that make the cancer cells less metastatic. Metastasis is when a cancer spreads from one organ to another. And the problem is for certain cancers, they are detected when they already did this process. And unless you can contain them, it becomes so much harder to treat. And so we're trying to stop them from spreading because that is an important thing. One of the major things that happen with current drug treatments is that patients become resistant to them. They stop responding. And so can we use marine natural products so that they act together with the current chemotherapy so you can use less of both and avoid this resistance? I'm an immunologist, so the most elegant way of eliminating cancer would be if you can get your immune system to respond to it. So we're trying to use marine natural products to restore that immune response. Of course, the best thing about cancer would be we can prevent it from ever forming, and so we're also looking at things that could prevent cancer formation in people that are at high risk. And of course, we're also trying to starve the cancer to prevent it from growing. And I'm going to give you some examples of how we do this in my lab. So, starving the cancer to grow. I'm going to go very basic. I'm sorry if this, but this is an easy way to think about it. So, all of us contain, or there are 4 sugars called nucleotides that come together to form deoxyribonucleic acid, or DNA. And this contains the whole map of who we are or what characteristics we're going to have. Whether I'm going to be short, tall, have blue eyes, brown eyes. And every single one of those sequences that defines one of those characteristics is called a gene. And we call genes that can be mutated and become cancers oncogenes. And one of the most common oncogenes that is often mutated in cancer is a gene we call RAS. RAS is mutated in about 30% of all human cancers. About 95% of pancreatic cancers have mutations in RAS, 45% of colorectal, and about 35% of lung cancers. RAS mutations are also thought to facilitate the growth of triple-negative breast cancer, which is a very aggressive form of the disease. So, we know that RAS mutations also activate unusual pathways by which the cancers get their nutrients. So could we starve the cancer to stop their growth? So we have a cancer cell with a RAS mutation. It activates these unusual pathways called macropanocytosis and autophagy. Yes, weird names. We love that weird names. So macropanocytosis is your cell basically kind of creates a little hat from its membrane. It makes it into a cup and brings anything that is outside inside of it to try to get nutrients that way. Autophagy is a little tough. So autophagy, another Greek word, means self-eating, and it is intended to be, if you were starving, your cell looks around what organelles you are not using, breaks them down, and uses that for energy. Cancer kind of has hijacked this system, and it grabs the healthy neighboring cell, breaks it down through this process, and that's how it gets nutrients. So we know, these cancer cells activate these pathways, and this leads to tumor growth. So could we use then a marine natural compound to block macropanocytosis and autophagy and stop that tumor from growing? And so here we are testing for that. I grew these cells and I took away their nutrients to promote micropanocytosis. And so these are pancreatic cancer cells. The nuclei is labeled in blue, the body of the cell is labeled in green. And I put a fluorescent sugar in the media. And so you can see how much of it has been internalized by the cells that they were trying to get their nutrients this way. And hopefully you will agree with me that when I use these marine natural compounds, you see a lot less red. There's less internalization of the sugar, and so we found inhibitors of macropanocytosis. For autophagy, we did slightly different things. So again, pancreatic cancer cells, nuclei is in blue, but now we're following a molecule called LC3B and a molecule called P62. And these molecules basically increase as autophagy is occurring, and they decrease as autophagy resolves. And that led us to find two different kinds of inhibitors. So you see this particular one, you see a lot less green. And so that stopped the process in its early stages, and we were excited about that. But then we had this compound called mansamin A, again, comes from a sponge, and you can see the accumulation of LC3B. So basically, this stops it at the very late stages. It doesn't allow it to resolve and sends the cells to apoptosis. So we're super excited about this compound. It has fantastic activities against pancreatic cancer. But I mean, all of these compounds have the potential of being potential cures, not only against pancreatic cancer, but for colon, pancreatic, breast, and lung cancers. And so we screened about 3,000 fractions for autophagy. We had two types of inhibitors, the early stage and the late stage. We got about 173 fractions from 70 organisms that were early stage inhibitors, and 277 fractions from 96 organisms that were late stage inhibitors. And 12 pure compounds have been isolated. So we're very excited about that. We have found a lot of them. And these are the organisms from which some of those fractions come. So, and for macropanocytosis, again, we screen about 3,000 fractions. We have 288 fractions from 123 organisms, and 3 pure compounds have been isolated with this activity. So we're in the process of further our understanding on that, so stay tuned. So now I'm going to tell you a story about preventing cancer formation. And we're focusing on molecules called advanced glycation end products. We like our weird names. And so these are compounds that form when sugars react with lipids, proteins, or nucleic acids, forming irreversible cross-links or bonds. And this happens anytime we cook our food at high temperatures, such as grilling, roasting, or broiling, or normal metabolic reactions in the body. Now, there are scavenger processes that are supposed to go look for these, eliminate them on a regular basis. But unfortunately, as we age, these scavenger processes stop working as great, and we start accumulating ages. And the accumulations of ages, because they function in so many different things, they help the movement of cells driven by chemokines, production of new blood vessels, oxidative stress, cell signaling, apoptosis. The accumulation of ages is associated with cancer, heart disease, Alzheimer's disease, and other diseases. And there are no currently drugs that bring down the bad effects of ages. And so, again, we know metabolism and diet lead to the production of advanced glycation end products or ages, and this leads to oxidative stress and inflammation, and that is the precursor of many diseases that are bad for us. Can we use marine natural products to block those ages, to stop their bad effects, and then stop the development of those diseases? So first we need to set up an assay, and so we started with human cells combining with a bunch of different classes of ages. We use our readout reactive oxygen species because we can follow that through fluorescence. Reactive oxygen species normally functions to tell your body that you're under stress. It's not a very good signal, so it's a bad thing to have. And so we also wanted to see, can we bring that down with our marine natural products? And so the first part is, can we follow the damage? So here I'm showing you the healthy cells in blue, and as you get mitochondrial, which is one of the organelles inside of cell, reactive oxygen species forming on it, you're going to see more red. And when you get just general reactive oxygen species, you're going to see more green. And you can see that as I gave more and more ages, I see more and more fluorescence. So that told me I can follow the damage caused by ages. So that brought to the cool question, the one that I really care about. Can we then bring that down, the damage, with marine natural products? And so here you have the solvent control. So that is just the methanol that we use to dissolve the compound. And you can see that it did nothing to the fluorescence. But we treat with a marine natural compound, and you see the fluorescence decrease. And so this particular fraction inhibited 78% of ROS and about 59% of mitochondrial ROS. So we're in the early stages of this grant. And so we established the assay. We saw that we screened about 500 of a small subset of our Florida library. We found about 5 pure compounds and 100 peak library fractions from which we can get new marine natural compounds, pure compounds. And so we're trying to identify the active compounds, and we're trying to use this data to get more funding so that we can continue this research. But we're very, very excited. I told you I'm an immunologist by training, and so why doesn't the immune system respond to cancer? Well, the reason is that cancer has become very, very smart in what receptors it has in the surface. And it either puts the ones that makes the T cell ignore it, or things that make it kind of invisible to a cell of the immune system. Could we restore this response? Can we make the immune system see the cancer again? So I apologize, super technical slide, but what I want to try to illustrate is, in order for a T cell of the immune system to recognize a cancer cell, you have a very orchestrated T cell receptor seeing the antigen in the concept of major histocompatibility complex. If this happens, you get the cancer cell dies, However, if this very orchestrated response happens in the presence of a molecule that we call an immune checkpoint, such as B7H4, and this one sees its receptor, what's going to happen is that cancer cell gets ignored. So an easy way to think about this, and let me see if I can do this.
We don't need to see his identification.
These aren't the droids you're looking for.
These aren't the droids we're looking for.
He can go about his business.
You can go about your business.
So for those of you who are Star Wars fans, you recognize Obi-Wan Kenobi using the Force to tell the guards to ignore the droids, right? Those droids are not the ones that they're looking for. And that's an easy way of thinking of an immune checkpoint, is Obi-Wan Kenobi using the Force to have the immune system not recognize it. So in this case, we don't want Obi-Wan, and we want to overwhelm him. And so one way we can do that is, can we block that B7H4 seeing its receptor and sending the signaling using a small molecule, thinking that should cause to restore the response and getting the cancer cell to die. And so this actually works in the clinic. It currently is being used right now. You might have heard of PD-1, PD-L1, or CTLA-4, and they're working beautifully. They are doing a lot of work against cancer. Unfortunately, there's cancers such as pancreatic cancer that don't express any of those. They express B7H4 or other immune checkpoints. So we wanted to focus on this one, B7H4. And so again, the cells are in blue, the body's in green. And what you see in red is that B7H4 molecule. And you can see how much of it is expressed in these pancreatic cancer cells. And so hopefully you will agree with me that we can bring it down using a marine natural compound. And so that should make it susceptible to be recognized by the immune system. We're also looking at another molecule called FOXP3. And FOXP3 used to be a marker for what we call regulatory T cells. So if you if your immune system ever mounts too hard of a response, it has its own recognizable T cells that go and say, hey, that's too much, bring it down. And so that's what the regulatory T cells are supposed to do. But a new finding is that cancer cells are expressing FOXP3, which used to be this marker for those special T cells, such as pancreatic cancer and colon cancers, and we don't understand how or why they use it. But you can see that they express a lot. So the red that you see here is that molecule FOXP3. And we think that they're using that to, again, evade an immune response. And again, you can see that when we treat with this fraction, we brought down the amount of FOXP3 that we see. So we're very, very excited about this finding because we have found marine natural compounds that bring down B7H4. And so those are potential therapeutics for pancreatic, colon, breast, and lung cancers. And those that bring down FOXP3 are potential therapeutics for pancreatic and colon cancer. And they might also be tools to understand why are they expressing this receptor. This is supposed to be a very tightly regulated receptor. They are not supposed to have it. So again, we're writing grants to continue this research, and so keeping our fingers crossed that we can get them. So the last story that I'm going to tell you has to do with 2D and 3D cell culture. And so this is how we grow cells in the lab. You know, we have them in a single layer against a tube or a flask. And this has provided us a lot of answers, and it's an easy way of doing it. But it has its drawbacks. So it limits cell-to-cell interaction. There are not nutrient or oxygen gradients present. And the extracellular matrix contact is mostly in one surface. If we do 3D cell culture or grow the cells as little spheroids or little tumors, now we maximize cell-to-cell interaction because they're talking to each other. There are nutrient and oxygen gradients present within the spheroid. There are better interactions with extracellular matrix, and this more closely mimics tumors. So if we were to screen our marine natural products using a 3D cell culture, those would be more clinically predictable that the compounds will work in the clinic. And so for this particular area, we focus on triple-negative breast cancer. This is a very aggressive form of breast cancer. It gets its name because it lacks the estrogen, the progesterone, and the epidermal growth factor 2 or HER2 receptors on their surface. And we have done a lot of progress against breast cancer by targeting those receptors. So those that lack all of those receptors do not benefit from targeted therapies. And they tend to be aggressive cancers. They're more likely to recur than other breast cancers. And they are what we call high-grade tumors. So there's minimal resemblance to normal cells. And so finding targeted therapies against triple-negative breast cancer remains an elusive goal. So we got a grant to do this effort. And we just finished with that work. And I want to show you some of the results because I think they're very exciting. So this is a spheroid under control. And again, here's going to be slightly different what we're looking at. The blue are going to be happy, alive cells. But now we use a marker that gets clipped when apoptosis is occurring. So the cells that are undergoing apoptosis are going to turn green. And those cells that are die or are dying, they lose their memory integrity, and they allow this red dye to go in. So you can see that in this control, at the center of the spheroid, I have some cells that are dying just simply because of those gradients. They are not getting enough oxygen. They're not getting as much nutrients as the cells that are outside of the spheroid. But hopefully you will agree with me that when I treat with a natural compound, I'm seeing a lot more cell death. And this is not, the story doesn't end up there. I know, super complicated slide, but bear with me. What I want to illustrate, so this is the 3D activity, and we follow them in 3 different forms, and within 24 hours, we see a lot of response. But I want to call your attention to the 2D activity and to the amount of compound that we're giving. So we went into very, very high doses. We treated for 3 days instead of just 24 hours, and you can see that I'm not seeing any activity. So this compound has selectivity for those cells that are in a tumor formation, meaning that they will attack tumors and not just any cell that they find. And this is another breast cancer cell line. You can see that the compound is more active in this cell line than it is in this one. And again, even though we saw some 2D activity it took a lot more of it to see it. So we use the IC50, which is the concentration at which we see 50% activity, as a measure to see the potential of any compound. And so you can see that in 3D, I have an IC50 of 1.5 micrograms per ml. In 2D, I have one of 98 micrograms per ml, and this is extrapolated just based on the data that I had. We couldn't reach that level. But that means it's 65 times more potent in 3D than 2D. So this is selective for triple-negative breast cancer as a tumor and not in 2D. Makes it insanely exciting because it means it will be very specific. So I told you that one of the major problems that we have with cancer is that our chemotherapies, people become resistant to our chemotherapies. And Taxol is a natural product, it's terrestrial, comes from a tree, is the drug that gets used the most to treat cancer, and it gets used to treat triple-negative breast cancer. But the problem is patients become resistant to it after a certain time. So what we did is, do we have synergy with Taxol? And synergy means the compounds work better in combination than either one of them does alone. And so we put our compound at its IC50, or that concentration that gives us 50% activity, then half of that and half of that. And we put Taxol in increasing concentrations. And so you can see the green, that means the cells are undergoing apoptosis, increasing. But hopefully you will agree with me that you see more green when we have the combination, and at the highest doses we see the most. But of course, use high content, it can quantitate, and so we get a slide that looks like this. This is a Taxol response. The best we got was 29%. We're focusing only on those cells that are undergoing apoptosis. This is the compound. We know about 50% because that's the dose that we use. Look at the combination of those. Now we're getting 98% of the cells undergoing apoptosis. And so this means that there is synergy between these two compounds and that we could use less of both to treat the cancer. So the last part that we wanted to test was, can we stop the cancer from spreading to other organs or the metastases? And so the way we did that is we took a spheroid, we gave it growth factors so that it would, one, expand. We put it in gel, and then we gave it media or media containing a natural product. And these are the results that we got. So you can see that my control expanded. So this is a way of me testing for metastasis in the lab. And now the marine natural compound. And so you can see that we reduce the amount of metastases that we're seeing just by treating with this particular compound. And this makes us excited because that was one dose, and so we can test that and optimize it and get it to stop the process altogether. So for the 3D spheroid cytotoxicity screening, we got an assay that identifies compounds that are active against triple-negative breast cancer cells when grown as a spheroid. We tested about 3,000 compounds, and we got about 150 fractions active in each cell line that we use. Out of that, 15 pure compounds have been isolated to date. 6 have been studied more closely. And for them, activity in triple-negative breast cancer or activity against cancer is a novel activity. Compounds are active in 3D, but not 2D. They all show synergy with Taxol. We have done proteomic work to try to determine how they work. And one compound decreases cell invasion in the lab. So again, we're trying to write grants. This grant just ended, so we're trying to get more grants to follow this work. But we're very excited because we have very selective compounds that are showing a lot of activity. And this is a Spongosaurus sponge, and that's dragmacidin D, one of the compounds that has that activity. Okay, so I just gave you a ton of science, more than you probably wanted to hear on a regular day, but I promise, if you remember the next slide, that's the most important slide in the whole presentation, and you will be okay. Okay? So are you ready? So are marine natural products great? So all of these organisms that I have here have marine natural compounds that might help us fight cancer. And so I'm almost done. I just wanted to, so whenever I give a talk like this, I always get 2 questions. What can I do to avoid cancer? And so we cannot do anything about our genetics. We cannot do anything about our luck. But the things that we can do to reduce our cancer risk is our eating a balanced diet, exercising regularly, maintaining a healthy weight, not smoking, avoiding excessive sun exposure. You need some sun. It helps us happy, produces serotonin. For those of you that are under 46, get vaccinated. The HPV vaccine prevents cervical, reproductive, and some oral cancers. So it's the easiest way you can fight cancer that you can find. And of course, getting regular checkups. None of us enjoy going to the doctor, but any cancer that gets found early is easier to treat than a cancer that is found late. So go to the doctor. The other question that I normally get asked is, how can I support your research? The most important thing is keep our oceans, our estuaries clean. We just had an expedition in April of last year to collect samples, and nothing makes us more sad than finding trash in the bottom of the ocean. And imagine, as I said, 70% of the Earth is covered in the ocean. What amazing drugs are there for us to find that we might kill before we even set in the effort if we're not good stewards of our ocean and our rivers and our estuaries. Of course, if you like what you heard, please tell others about the research that we're doing and why it matters. I always like when people know what we do. It's important for us. We depend on funding to do our research and to keep our jobs. So again, if you like what you see and you hear that the NIH or NOAA are going to lose their funding, write your representatives and tell them, please don't do that. We also get a lot of funding from the Florida Biomedical program. So again, similar thing, please support that. And of course, if you feel like it, make a donation. You can choose from the Florida Atlantic Foundation website, choose pancreatic cancer research, or you can scan this QR code. And the reason we use donations is that we have very set guidelines in our grants of what we're allowed to do. So whenever we want to pursue a new direction, do this experiment that's a little offhand to see if it works, those donations help us do that. So I just want to acknowledge our sources of funding that include the Florida Biomed, the National Cancer Institute, the state license plate that are granted through the Harbor Branch Oceanographic Institute Foundation. We have a couple of local foundations that support students in the summer to work in our labs. We get samples through NOAA Ocean Exploration, the Cooperative Institute for Exploration, Research and Technology, and NOAA Deep Sea Coral Research and Technology. And of course, the Harbor Branch Oceanographic Institute gives us seed funding to test some of our crazy theories. And the Bernard A. Egan Foundation is a local foundation in Fort Pierce that has supported my research for years. And of course, we're a small group in the Marine Biomedical, so chances are that the work that I show you involves somebody there. And I particularly want to highlight my technicians, Deidre Harmony and Tara Pearson, in particularly Tara, because the bulk of the work that I show you was done by her. And with that, I'll take any questions. Okay, there was a question right over there.
Esther, thank you. Dr. Guzman, thank you so much for that amazing talk. Everybody, right? Wasn't that amazing? Before questions, I'd like to introduce Jennifer Dillon, Executive Director of Development, and she's going to host the Q&A session.
Thank you. Thank you so much, Esther. That was wonderful, and I get to follow a great speech. I'm not solving cancer, but I am raising money for our students. And if you'd like to support any students that come to the University of Miami, we have lots of students who are doing great work here. I encourage you to fund either our a general scholarship or our opportunity fund, or if you'd like to help us support our new center, I would welcome that as well. My name is Jennifer Dillon. My email right here. You can connect with me. And while I do that, I'm just going to go ahead. I don't want to take any more time up of your questions. Seems like we have a lot of very interested people. So what I'm going to do is raise your hand when you want to have a question. We'll send our students, these are our student volunteers, up to you. You'll get the microphone. Ask your question. Please hand the microphone back to them. And we'll start with questions. Thank you.
Hi, thank you for a great presentation. So a question. All of these commercial products, I just want to clarify this, or I want you to clarify this. Are they produced from the organisms that are taken from the ocean, or in large quantities, and do you extract substances from them, or do you synthesize them? I think I know the answer, but I think we all need to know.
No, that's a fantastic question. And so you just hit the nail on its head. One of the major problems, if I find one of these great compounds, is that they come from deep sea. And so we used to have the submersible that went all the way to 3,000 feet. Now we don't have any tools that go that deep. And so we cannot just simply go to the ocean and grab another sponge and grab many other sponges. So we have different ways in which we try to make them. So the first solution is you find the structure and somebody makes it in their lab. Some of the structures are very amenable with that, and if that happens, that's the easiest route that we get. There's some compounds that are made by the bacteria that live inside the sponge and not by the sponge itself. So for that, we have scientists that look into the bacteria DNA, and they can follow basically, like I told you what a gene was, they follow those genes that are responsible for making a compound. And the holy grail is we're going to be able to get those compounds or those genes, put them in something like E. coli that grows so fast and so well, and get them through that. Even if we just find the bacteria that makes it, we could ferment it and get it from that. One of the advancements that is happening at Harbor Branch right now is that they're trying to create sponge cell cultures. So in our last expedition, we got sponge cells frozen so that they can be cultured in the lab. And so if we found a compound that comes from one of those sponges, they can grow the cells in the lab and now make it in the lab using the sponge cells. So we have many different ways in which we're approaching that, because yes, it is not feasible to just go back and try to get many more sponges and use that as a source. Oh, and I forgot to say, aquaculture is sometimes another source. We have tried to grow the sponge in like aquaculture, and so that's another way that we can go around. Hi.
I was wondering, how do you or anybody, like scientists, think to look at specific organisms in certain deep environments?
So the whole story of Harbor Branch is that somebody came up with the idea of using the sub, and they were saying, okay, so we're using this for ocean exploration, we should look at this for drugs. And that's really how it happened, and it started in 1984. And there's a lot of history of using nature to provide those drugs. It all comes from, terrestrial compounds, but again, there's so many similitudes, there's so many different organisms that live in the ocean that it just kind of made sense that if we're getting, taxol from a tree, there could be things in a marine organism that could help us find those, new drugs. So I think it is kind of like any science question, it is always based on what somebody did before. Somebody went, and everybody knows that, people in ancient times used to use plants, they got a toothache, they cut the bark of a tree, they took that, and that's how we got aspirin, and aspirin is the modified, synthetic, made in the lab version of a natural product, and that made it more stable, and so we can always carry in our purse. Versus the bark of a tree stops working after a little bit, And so I think we build on the knowledge of people that came before us.
Fantastic presentation. So
Thank
just
you.
to add, kind of carrying on what you just said about building on knowledge, we live in a big world. There's other scientists and doctors doing the work that you're doing. So with the introduction of AI, and you didn't, I don't know, like accelerating your work, connecting your work. You shared an example early on where something in the 60s took, the life cycle of that whole process was 30 years. So I don't know if you're thinking about or could talk a little bit about accelerating this fantastic work.
Oh, absolutely. I mean, so simply, I told you I love my high content imager. And we're looking at, do we see a reduction of the red or the green? But there's so much more data within that. So we sometimes change the phenotype, sometimes we check the nuclei, and we're not looking at all that data. Having something like artificial intelligence being applied to that will allow us to look at those, because I told you, I use 15 plates of 384, no human can see it, and look at those images. And we take 9 images per site. And so for somebody to go and sit and look at that, it would be impossible. That's where we would use machines to learn. There's also collaboration. So we're scientists. I mean, to me, if one of my drugs, like Mansamin A is a perfect example of that. I told you that that compound has a lot of activity against pancreatic cancer. So we published that work, we presented that work to conferences, and now another university is pursuing it for a different type of cancer. And I tell you, if it makes it, I'm gonna be elated because I consider a compound my baby, and all I wanna see is making it to the clinic. And so I think we also collaborate with other people. We try to we have had collaborations even with pharmaceutical companies, Novartis and SI Pharmaceuticals, to try to bring things closer to the clinic. And so for us, it's really important to advance the knowledge, and if AI is a means to do that, we're all willing to do it.
Yes, I'm interested by the apoptosis.
Apoptosis?
It looks like a Greek island where I would like to go on holidays, but I understand it's when the cell that, some cells don't want to go to apoptosis. They don't want to die, right?
Some cells don't want to die, but it is a normal process. So you use apoptosis even through development.
Have we studied sufficiently apoptosis to see if, the contrary, the fact that cells don't want to die, if we could apply this to all the other cells of the body, that would be kind of nice.
Well, it's a two-sworded, a two-edged sword, if you think about it. So one of the characteristics of cancers is that they don't want to die. And the reason you're getting the signaling to undergo apoptosis is because those cells are damaged. And so we need to eliminate them because that will be the easiest way to do it. And so, yes, if we want to live forever, it might be good, but in what conditions do you want to live? Because, having cancer might not be the best way of having all your cells respond. So apoptosis, you need it to occur. There's a lot of, so we use mice that we knock the apoptotic genes off to try to prevent apoptosis from occurring. And they develop lymphadenopathies. They have all these different diseases because apoptosis is necessary to maintain balance in the body. So your body always want to have a balance. You need certain things to die, certain things to be alive in order for you to be healthy. And the minute you eliminate one of those processes, you cause disease.
Thank you, Dr. Guzman. I want to say tonight's presentation will be available in the coming days on our Facebook and our YouTube channel. And we would like for you to join us on the 25th of February. We're going to have another lecture that evening. It's our third one. Thriving with Fire is the title of it. Very timely, and it's featuring Dr. Michael Falkowski. Thank you. Thank you so much. Have a good night.